All right, so today we're going to talk about the first little bit of inheritance in the study design, really. And it's actually the very last little bit of the study design. So, this is the simple genetic cross, and this is stuff that we did in Year 10 science, but we're going to go into it a little bit more detail. The scientific name, by the way, of the simple genetic cross is the monohybrid cross. All right, pause the video here, have a go at these three quick questions, and we're going to go through them together in class, okay? All right. So in the study design, um, we're going through Unit 2, Area Study 2. So pedigree charts, we're not going to go through that part. Genetic cross outcomes, we'll talk about that. And genetic decision making, we'll talk about a little bit. And then also pedigree charts and the patterns of inheritance, including autosomal dominant, autosomal recessive, X-linked and Y-linked traits. And then also the determination of genotypes and the prediction of the outcomes of genetic crosses. So the outcomes, we mean the phenotypes, okay, including monohybrid crosses and the monohybrid test cross, which is a specific kind of monohybrid cross. All right, so today we're going to discuss how did Mrs. McKinnon get red hair. Okay, as you can see in this lovely picture of Mrs. McKinnon, she has red hair. But because the story is a little bit more complicated than that, and that is something very important that I'm going to talk about, is that this is an oversimplified example, okay? Red hair is something that we call polygenic, which means that it's controlled by lots and lots of genes, hair color, but monohybrid or simple genetic crosses are actually something that we can only talk about in terms of monogenic inheritance, which means that there's only one gene. Okay, regardless, I want you to follow along today and use your show me boards to actually have a go at doing a few of these examples. And as you go and do them, do call me over because I want to check that you know how to do them. All right, before we talk about any of this stuff, you can't talk about the monohybrid cross without talking about this guy, Gregor Mendel, who was a monk, an Austrian monk back in the 1800s, and he actually discovered a lot of these concepts of genetics through his experiments making peas have sex, okay? And in his results of his experiments are shown here, and he actually found out the inheritance of flower color, flower position, seed color, shape, pod color, stem length. All right. And he found out that all of these traits in peas are all due to simple genetic crosses. Okay. By the way, if you ever see a P like this, that's because we're talking about the paternal. And then the future generations are the F represented by this F. So F1 is our first generation after the paternal, right? And then F2 is the second and third and fourth, and so on and so on. All right, so before we talk about any inheritance, I want you to have a go at doing this, okay? So Mrs. McKinnon's dad is an individual with the dominant phenotype. He has black hair, all right, okay? He produces gametes through meiosis, and we want to represent the division of his gametes through meiosis, okay? I'm going to give you a little bit more information because I know that even though Miss McKinnon's dad is an individual with the dominant phenotype, black hair, the only way we're going to have Mrs. McKinnon have red hair is if he is heterozygous. Okay? We're going to make our lives nice and easy and identify that he is heterozygous and he actually has the big H, little h allele. The big H, little h genotype. Okay? All right, so let's make some gametes. So initially, Mrs. McKinnon's dad has a mother cell, and this specific mother cell is from his germ line, which just means that it is in his gonads. Okay, it's going to produce spermatozoa. Okay, and inside of that, we're going to oversimplify it, and we're going to show two chromosomes Okay. And those two chromosomes are actually going to have the hair color allele on them. One of them's going to have big H, and one of them's going to have that little h. Okay? All right, let's do this. So, what's the first step in meiosis? We have meiosis 1. Okay? And oh, hang on. We don't have meiosis 1 yet. What do we've got something wrong there. We need to actually we can put meiosis 1, but first we need to duplicate these chromosomes very neatly, just like I am there. Perfect duplication. All right, now let's do this. Meiosis 1. Okay, and meiosis 1 is going to produce for us 2 daughter cells. Okay, and each of these daughter cells are actually going to get one of these homologous pairs. Right? And there's the allele on each one. And now remember that both of these sister chromatids, they both have this big H. So that means that this big H is actually going to go to both of these. And same on this side, both of these are going to get the little h. Okay? Now let's show meiosis 2. So meiosis 2 is going to be each one of these is going to divide further. All right, here's meiosis 2. Okay, and then we're going to end up with obviously these are all the same size, just I'm getting stuck for space. And each one of these are going to have a single linear chromosome. All right. And if you look, these two that are on the left, they're going to have the big H allele, and these two on the right are going to have the little h allele. The last little bit of steps that's going to happen is because we're producing sperm, we're going to go through the process of spermatogenesis, which just means out of these we're going to make some sperm. Okay, that's what this is called, sperm. But regardless of that spermatogenesis, the important part that we want to look at now is that we have these gametes, and these gametes either have the big H allele being carried or the little h allele being carried. All right. The same exact thing is going to happen with her mum. All right, and her mum was also big H, little h. She's also heterozygous. All right, so what were the chances that she had red hair? Let's talk about this monohybrid cross. Okay, so both of her parents have found to be heterozygous, like we said, for the hair color gene, found on an autosomal chromosome. All right, we're going to produce a Punnett square to show this cross, and we're going to state the chance of having a child with red hair. Okay, before we do any of this, we need to identify the alleles. All right, and we did that before, but let's say the big H was black hair, and the little h is red hair. All right, now that we've identified alleles, all right, we're going to place onto a Punnett square, and how we place this is, it doesn't actually matter in terms of the parents, but pretty much we're going to have the parent 1 gametes possibilities. We're going to put that on the top. So remember that they're both heterozygous, so big H, little h, because they're the two gametes we can get. And then on the other side, we're going to put in the parent 2 gamete possibilities. Okay, and same thing, we've got big H, little h. Okay, and then to do these crosses, in this first box here, okay, all you're going to do is grab these two gametes like this. So big H, little h. The next box, same thing, this gamete and this gamete. Remember that always put the big letter first, all right? This gamete, so big H, little h, and the last two little ones, little h, little h. Okay? Now, I draw this little data table. I just find it actually helps me organize my results. You don't have to, okay? But I do find it actually really does help. All right, so let's have a look at the results. What are the possible genotypes? Oh, I've made a mistake. Big H, big H. What are the possible genotypes? So the first one is this big H, big H. The second one is the big H, little h, which we also have here. We don't have to write that twice. And the third one is little h, little h. Okay? Now, what are the possible phenotypes we can get out of this? Hopefully, you've identified that both of these genotypes are both going to give black hair. This one on its own is going to give that red hair that Mrs. McKinnon got. Okay? And then we can actually further analyze this data using percentages. Okay? So, in terms of percentages, we've got 3/4 or 75% having black hair, and a last quarter or 25% having red hair. Okay. The last thing that we're going to talk about is the ratio. Okay. The ratio in terms of the monohybrid cross is very, very simple. Okay. In this case here, we have 3 with black hair, 2, 1 with red hair. Okay. I'm generally going to ask you for percentage, but you might find some questions that do ask about ratios. All right. So I'm going to let you having a go at the monohybrid test cross because Miss McKinnon's sister believes that because she has the dominant phenotype, black hair, it will be impossible for her children to have red hair. All right. Her new man, Bruce, has actually got red hair. All right. We want to prove her wrong using the test cross. And what you're going to cross to show the test cross, okay, what the test cross is the homozygous recessive, which in this case is Bruce, he's our test cross, and that's crossed with an unknown dominant. Okay, so in this case, we're going to be crossing Mrs. McKinnon's sister, we know has black hair, so she has at least one big H, but the other one we don't know. We're just going to put a dash there. And she's crossing with Bruce, who is little h, little h. Okay? Have a go at doing this. Have a go at seeing what you've got, and we'll discuss this together in class. All right. Last little example, I want you to try it yourself. We have 2 homozygous recessive individuals with blue eyes are being crossed. We're going to use a Punnett square to show that their offspring must have blue eyes. Okay. Identify alleles first. All right, and then have a go at doing the Punnett square. And lastly, have a data table to show the results. This is probably the easiest little cross that you can do. All right, so that's monohybrid inheritance on autosomal chromosomes. Okay, so these are the autosomes, but we can actually talk about monohybrid inheritance in terms of the X chromosome and the Y chromosome, so the sex chromosomes. And a great example of this is hemophilia A. What hemophilia A is, it's a horrible genetic disorder in which individuals have a defective blood clotting protein. Okay, and hemophilia A is recessive and is found on the X chromosome. So a male with hemophilia is crossing with a female without hemophilia A. Okay, and she actually has no family background of hemophilia A. When you get that no family background of hemophilia A, we can quite confidently we can quite confidently assume that in this case we actually have a homozygous dominant individual. Okay, because she doesn't have. It's very unlikely that she'd be carrying without any family background at all. Okay, we're going to identify the phenotypic percentages of this cross. Let's do the ratio as well. Why not? All right, so let's identify alleles. When we're identifying alleles for X-linked inheritance, it works slightly different. Okay, so obviously we're talking about the X chromosome and the Y chromosome. The Y chromosome doesn't change. Okay, so you can just ignore that for X-linked inheritance. But for the X chromosome, what we're going to do is write a little a at the very top. Well, a big A in that case, or a little a like this, okay? So hemophilia A is recessive, so this guy here would have hemophilia A. Okay, and if you have the big A, that's the allele for the normal gene. Okay, let's do the Punnett square. So remember, at the top we're going to. Oh, actually, before we go on to the Punnett square, what's the actual cross in this case? So we have a male with hemophilia. Okay, so he actually has to be X little a Y. All right, and we're crossing him with a female with no family background. So X big A, X big A. Okay, so now let's do the female up the top here. So the female. What are the possible gametes out of here? So we've just got the two X big A's. So X big A, X big A, and then let's do the male on this side here. You don't have to write male, female, just doing it so we can see it. X big A and Y. Okay, and then we're going to do just like a normal Punnett square. So X big A, X little a, X big A, Y, X big A, X little a, and X big A, Y. Okay, what are the two possible genotypes? So we have X big A, X little a, and we have X big A, Y. Okay, and then what are the phenotypes here? In terms of X-linked inheritance, it is quite handy to actually identify male and femaleness. Okay, if the question doesn't specifically ask for it, you don't have to. But I'd say you're always better off just doing it. Okay, so for example, in this case, we have a female who is normal. And in terms of phenotype, that's all you have to write. But she is also what we call a carrier, okay, which is another way of saying she's heterozygous. In this case here, we have a male who is normal, but he is not a carrier, okay? Percentage-wise, it's 50-50. And for the ratio, it's just 1 2 1, okay? All right, let's do another one. This is Y-linked inheritance of the SOX21 gene. So what the SOX21 gene is, it's found on the Y chromosome of humans, okay? And the gene has two alleles. We have the bold version and the normal version, okay? A bold male mouse crosses with a female mouse with normal hair, okay? We're going to use a Punnett square to describe the possible offspring of these two individuals. You're going to see that Y-linked inheritance. It's a funny one because it's actually really easy, okay? So the alleles, we either have bold or normal. In Y-linked inheritance, I'm going to write it like this. So YB is going to be bold, and then YN is going to be normal. All right, why I use two different letters there is for this reason down the bottom that we'll discuss in 2 seconds, okay? Let's have a look at what the Punnett square would look like. So let's go female at top again. So we're crossing a female who's XX with a male who is bald. So X, Y, B. Okay, so XX with X, Y, B. And hopefully that's what you get. So here's the genotypes, XX, X, Y, B, and the phenotypes, female. There's no Y chromosome, so we have to make no judgment about whether it's bald or normal, but so we just write female. Oh, and then here we just write male and bald. Okay, percentage-wise, we have 50% female and 50%, and ratio is 1:1 again. Okay, why can't we describe Y-linked traits as dominant or recessive? Is because there is only one version of the gene present at any time. Okay, we've only got one Y chromosome in males. Females don't have one at all. So it's not like you can go and turn around and compare YB with YN. Okay, you're never going to have this genotype. It's not existent. Okay, so that's why there's no recessiveness and dominance in terms of the Y-linked inheritance. Last one, have a go at doing this little example of Rett syndrome on the X chromosome, and make sure you do show me on your show me boards.